Good morning, everyone, and thank you for joining us on this call today. We are delighted to share with you today the positive preclinical data from VBI 2900, our prophylactic coronavirus vaccine program, which enabled us to select two optimal candidates to take into an adaptive phase 1/2 clinical study, expected to begin around year-end 2020, subject to regulatory approvals. I would refer you to the press release we issued this morning for more details and for disclaimers regarding forward-looking statements, which are also outlined here on the slide. For additional corporate information and detail, I refer you to our SEC and our Canadian filings. In March of this year, we announced a collaboration with the National Research Council of Canada to develop a vaccine program to address the coronavirus pandemic that has continued to have significant public health, economic, and societal consequence. As part of this collaboration, a series of 3 preclinical mouse studies were initiated in the second quarter of 2020 to enable the selection of optimized vaccine candidates to advance into clinical trials. These studies looked at the impact of VBI's proprietary enveloped virus-like particle, EVLP platform technology, on the expression of the SARS-CoV-2 spike proteins compared to standard recombinant expression. Differences in conformation of the SARS-CoV-2 spike protein, and finally, the benefit of a variety of adjuvants. Coronaviruses are members of the enveloped class of viruses, which make them prime targets for our flexible enveloped virus-like particle platform technology. Multiple EVLP candidates have preclinical and clinical proof of concepts and have shown to be highly immunogenic because of their structural similarity to viruses found in nature, customizable, safe, containing no infectious genetic machinery, and have been proved to be safe and well tolerated in other clinical studies, and commercially viable. EVLPs are manufactured and purified using highly scalable methods that can leverage existing capacity at contract manufacturing organizations. As a result of these studies, the data from which we will discuss shortly, we selected 2 vaccine candidates to advance. VBI 2901, a trivalent EVLP, co-expressing a stabilized, modified pre-fusion form of the SARS-CoV-2, COVID-19 spike protein, in addition to the SARS and MERS spike proteins. VBI 2901, a monovalent EVLP, expressing a stabilized, modified pre-fusion form of the SARS-CoV-2 spike protein. As the data to be presented on this call will demonstrate, we believe that the results from these preclinical studies that after just 1 dose, our vaccine candidate are equivalent or even better than other published preclinical and clinical candidates after 2 doses. Pending further clinical development and regulatory feedback, we believe that our candidates have the potential to be a single-dose vaccine. So with that, I'd like to turn it over to VBI's Chief Scientific Officer, David Anderson, PhD, a Harvard-trained immunologist with over 20 years of experience to discuss our data. Over to you, Dave.
Thanks, Jeff. Good morning, and thank you all again for your interest in VBI and our coronavirus vaccine program. Having worked so closely with all those involved in the design and execution of these preclinical studies, on behalf of the R&D teams at both VBI and the National Research Council of Canada, the NRC, we are excited to share these data with everyone today. As we've seen with data from other COVID-19 vaccine approaches, comparison to convalescent sera is an important component of early data. It enables a benchmark for immunogenicity and potential efficacy, and also enables comparisons across programs. On slide 4, you can see that we sourced a panel of convalescent sera from 20 individuals who had contracted and recovered from COVID-19. Consistent with observations of other scientific groups, variance in antibody responses exists among COVID-19 recovered individuals. As you can see in the bottom left-hand graph, we collected samples from donors who mounted robust or high titer antibody responses, and from those who had weaker, low titer antibody responses. Others in the field have demonstrated a correlation between antibodies which bind to the COVID-19 spike protein and those with functional or neutralizing activity. As you can see in the graph at the bottom right of this slide, high titer sera did indeed demonstrate higher neutralizing antibody activity. To quantify neutralizing activity in all of these studies, we used a plaque reduction neutralization test with a stringent 90% inhibition threshold. It's called PRNT90. This represents the extent to which sera can be diluted and still prevent virus infection by 90% or greater. You will note that the low titer sera do not possess neutralizing activity that can neutralize greater than 90% of the virus in this assay. The use of high titer convalescent sera and a PRNT90 threshold sets a high benchmark for our program, but we believe it is important to rigorously assess the vaccine candidates in order to have a better sense of the potential immunogenicity and efficacy in clinical trials. Slide 5 demonstrates impressive induction and boosting of neutralizing antibodies, which are the presumed correlate of protection against COVID-19, using a panel of vaccine candidates. The two black bars represent the native form of the COVID-19 spike protein, with and without a modification, and how it is expressed on the surface of VBI's EVLPs. You can see induction of significant neutralizing activity, well above high titer convalescent sera, but only after two doses. The blue bars represent neutralizing activity with an optimal or pre-fusion form of the SARS-CoV-2 spike protein. First, comparing the striped blue bar with the open blue bar, you can see a clear benefit of EVLP expression versus a recombinant protein. The recombinant pre-fusion spike protein only induces neutralizing activity above high titer convalescent sera after the second dose, and even then, it's the weakest of the group evaluated. Taking the same spike protein, but expressing it on the surface of VBI's EVLPs boosts functional neutralizing activity above high titer sera after the first dose, further increasing it after the second to a 64-fold higher titer. Finally, represented by the solid blue bar, we made a modification to the way the optimized pre-fusion spike protein was expressed on the surface of the EVLPs, and this induced neutralizing activity after only one vaccination, a titer that was 4-fold higher than high titer convalescent sera, increasing to 64-fold higher after a second dose. Not included on the graph here, we tested a variety of adjuvants, including alum phosphate and TLR-based adjuvants, that further improved the induction of neutralizing titers by approximately 5-fold, and also promoted strong Th1 type antibody and T-cell responses, which may de-risk concerns about potential for enhanced disease and associated lung pathology. Slide 6 builds out the data, demonstrating antibody binding titers after just a single dose of vaccine, which further emphasizes the benefit of EVLP expression of the optimized pre-fusion form of the SARS-CoV-2 spike protein. The far right column illustrates an antibody binding titer that is 10 times higher than both the high titer convalescent sera pool and antibodies induced with the recombinant pre-fusion protein. Based on the data on this slide, in addition to the neutralizing antibody data illustrated on the previous slide, we believe our optimized vaccine candidates may offer rapid, potent protection with the potential to be administered as a one-dose vaccine. Having demonstrated the importance of EVLP expression of an optimized pre-fusion form of the SARS-CoV-2 spike protein and associated immunogenicity, slide 7 illustrates the breadth of immunogenicity induced by VBI 2901, the trivalent pan-coronavirus candidate. Not only was the vaccine able to induce antibodies against SARS-CoV-2, SARS, and MERS, but the vaccine also induced antibodies against human coronavirus OC43, one of the most common circulating seasonal coronaviruses, which is associated with common colds. The induction of this response to OC43 demonstrates exactly what we were hoping to see with the trivalent approach. That is, broadening of immunity to other structurally similar coronaviruses not expressed within the vaccine. COVID-19 is an RNA virus, like influenza or the related SARS coronavirus, and as such, it has the potential to mutate over time. While SARS-CoV-2 has not mutated as quickly or drastically as influenza or SARS in the past, we know that there is already some degree of change. The predominant strain circulating in Europe differs from the original strain that first circulated in Wuhan, China. The broadening of immunity to OC43 suggests that the vaccine may also have broadened immunity to potential new COVID-19 variants that may emerge over time. I will caveat the data displayed here. This data used an unmodified, unoptimized native form of the SARS-CoV-2 spike protein, and as such, the data presented is after 3 doses. However, the breadth of response we see, even with an unoptimized candidate, leads us to believe that the expression of the optimized spike protein in the trivalent candidate, as well as the potential inclusion of a potent adjuvant, would further increase antibody binding titers and neutralizing activity after just 1 dose. With that, I will hand it back to Jeff.
Thanks, Dave. Before opening up for questions, I'd like to take a moment to thank our partners at the National Research Council of Canada, as well as every member of the VBI team. It is with thanks to their intense dedication and drive through these truly unprecedented circumstances that we are able to share these exciting data with you today. These early data have demonstrated the induction of robust neutralizing and binding antibody activity after just 1 dose. And as Dave just discussed, the trivalent construct VBI-2901 may offer additional benefit in the form of increased breadth of reactivity to protect against a wider range of related coronaviruses. To reiterate, we do believe these preclinical data after 1 dose demonstrate equivalence to or improvement over other published candidates after 2 doses. As such, depending upon results observed in the upcoming clinical studies and subject to regulatory feedback, we believe these candidates may be viable single-dose vaccine regimens. In early August, we announced an award of up to $56 million from the Canadian government, which we are extremely grateful for their support for, to accelerate and develop and support development of these 2 candidates through phase 2 studies. Further discussions with the Government of Canada may explore additional federal funding to support late-stage development. To support the clinical development, we also recently signed a manufacturing development agreement with Therapure Biomanufacturing, a CDMO located outside of Toronto, Ontario. We expect Therapure to initiate GMP clinical manufacturing in September to enable the availability of clinical materials by Q4 2020. Moreover, as part of this partnership and upon successful clinical outcomes, there is a potential to leverage capacity more broadly to support large-scale manufacturing. We expect to initiate an adaptive phase 1, phase 2 human clinical study around the end of the year. In this study, based on the preclinical data seen today, we expect to test human doses in the range of 2 to 5 micrograms. A low dose we believe is made feasible by the improved immunogenicity of our envelope virus-like particle technology, as well as the further optimization of the COVID-19 spike program. With that, I will now turn over the call to Nicole Anson, VBI's Director of Corporate Communications and Investor Relations, so that we may take your questions. Nicole?
Thank you, Jeff. We will now open the line for any questions. If you would like to ask a question, please use the raise hand button at the bottom of your Zoom screen. Please hold while we pull for questions. The first question will be from Steve Seedhouse of Raymond James. Steve, please go ahead and unmute your line at this time.
Hi, can you hear me?
Hey, Steve, we can hear you.
Yeah, we can hear you, Steve.
Oh, great. Thanks, guys. Congrats on the data. Thanks for taking the question. Good morning. Just wondering if you could talk about the kinetics of the neutralizing antibody titers, when they emerged, how long they lasted, and if you saw them continuing to increase over the course of the assay. And then also wondering if you could comment on, just elaborate on CD8 T cell response.
Yeah, Steve. So in terms of kinetics, we don't have a sense of the durability yet, so I can't tell you about that because we're still following the mice. In terms of how quickly neutralizing antibody occurred, we looked 14 days after the first immunization and then 14 days after the second. So within 2 weeks, yeah, after a single dose, very rapid onset. We did see very good boosting between the first and second doses, so on the order of 10-fold boosting. And in one of the studies, even after a third dose, you saw the same thing. So very rapid onset. I'll have to get back to you as the data emerges in terms of the durability. In terms of CD8s, we're looking at now. We've used an ELISpot assay. We know we induce gamma-secreting T cell responses, but haven't started to fractionate to understand whether it's CD4, CD8 dominated, or a bit of both.
Okay, appreciate that. Thank you. And then clearly you have evidence that you're meeting your goal here of broad activity given the trivalent nature of the vaccine, which is obviously a unique approach you guys are taking. I'm just wondering if you could elaborate on the implications of that. Obviously, in terms of how you would ultimately position this vaccine, but also just in designing the clinical development program and whether you would focus on the same endpoints that the other COVID vaccines are focusing on, or whether there would be some different metrics to look at the broad activity that you would target in the clinical program.
Yeah, let me, excuse me, let me answer that, Steve. I think first and foremost, we're in detailed discussions with Health Canada at this point in time around what the phase 1/2 trial looks like. But we certainly will be taking both into the clinic. And you may ask why. First and foremost, because in terms of the production process and in terms of yields and volumes and everything else, the monovalent is likely to be more efficient in the manufacturing process because you just have one antigen rather than three. But we do believe that over time, we will see mutations in COVID-19. And as such, we do believe that in the midterm, the trivalent is a very attractive vaccine, particularly for people who may travel extensively and be exposed to slightly different strains. As Dave mentioned, we see a slightly different strain between Europe and the US at the moment, or at least Europe and China and the US at the moment. And of course, SARS and MERS have not gone away. MERS is still prevalent in the Middle East, and SARS is still circulating in Canada and in China. Sorry, and there is, obviously intense public health concerns about the reemergence of SARS. So it may be, Steve, that our monovalent is relevant for a rapid launch and particularly focused on COVID-19. But certainly shortly behind that, I think the trivalent is going to be extremely relevant in tackling the public health challenge globally that coronaviruses face.
Yep, understood. Okay, well, thanks for taking the questions, guys, and very nice data.
Thank you.
Thanks, Steve.
Okay, and we have a written question from Jameson Cow over at BMO, and it's also from Do Kim. How important is the higher neutralizing antibody activity with one dose of modified prefusion spike protein?
Yeah, I'll let Dave answer that scientifically at a moment, but I think I can honestly and truly say that VBI team were blown away when we saw these remarkable results. And it really does prove the benefit of an envelope virus-like particle platform against an envelope virus. And we were blown away, but perhaps not surprised because it is logical. And we do see that the subunit vaccines, generally as a class, are significantly more potent than either modified RNA or other RNA platforms at a much lower dose. We should recall that we are planning human doses of between 2 to 5 micrograms. Other approaches, whilst completely valid, of course, are at 100 micrograms at 2 doses, which produce lower neutralizing antibodies than 1 dose of our vaccine. And that is comparing mouse-to-mouse data from previous published mouse data by some of the other approaches. Now, of course, for the sake of humanity, we believe and we hope that some of those leading candidates, which are ahead of us in the clinic, will be successful. But having said that, I really do believe, given that we do not understand the kinetics of COVID-19 and the potential mutations, that there's room in the market for many different approaches, particularly one that would be at a lower dose and potentially a significantly lower cost of goods. Now, with regards to the benchmark against convalescent sera, I think Dave and his team, as always, have taken an incredibly rigorous approach by accessing this bigger panel of convalescent sera, both high and low. I'm not saying others have done it, but you can pick against a low benchmark and you'll look more remarkable. And we've used the most stringent assay, the PRNT90 assay. In the early days, some of these results were published against a PRNT50 assay and then went to a PRNT80 assay. We're now a PRNT90 assay, which really is most stringent. But from an immunology perspective, I'll let Dave answer the significance of the degree to which we have exceeded that GMT of the high convalescent serum benchmark.
Yeah, so I think when you think about what the benchmark represents, it supposes that naturally acquired levels of immunity will protect against disease. And so that's why it's established as a threshold. But that level isn't typically 100%. It may be, say, a 70 or 80% protection rate. And so if you can surpass, that benchmark established with convalescent sera, the hope is that not 75 or 80% of individuals that receive the vaccine, but closer to 100% would get to protective levels. So that's one way to think about, the desire to be higher than the convalescent sera benchmark. But beyond that, the higher the antibody titers, the longer the durability of that response. Antibody responses will typically always decay over time. And it just kind of makes sense that the higher the titer, the longer it's going to take for those levels to decay below a protective threshold. So 2 different reasons we think being as high above the benchmark as possible is of great importance.
Great, and we have 2 final questions from Jamison and Doe at BMO. The first, Dave, you just may have spoken to, but to reiterate, what is the titer hurdle for a 1-dose vaccine? And 2, were the antibodies generated against OC43 neutralizing?
Yeah, so to answer the first question, if you look at the highest titer convalescent sera using the PRNT90 assay, that titer was 1 in 40. If you looked at the optimized monovalent candidate, the titer there was 1 in 160, so 4-fold higher, which is certainly significant and outside any kind of assay variability. In terms of neutralization against OC43, we're still in the process of trying to run those studies. As you can imagine, there's a huge crunch and prioritization towards getting neutralizing assay data for the primary target, COVID-19. We will be, are in the process of trying to ask that question against OC43. It's clearly important. As we've said before, we want to make sure that we're not just inducing antibodies against COVID-19, MERS, SARS, OC43. We need to know that those antibodies have functional neutralizing activity. And that's data that you can expect to see in the future in a publication that we're working on right now.
Great. This concludes our question and answer session. I would like to turn the call back over to you, Jeff, for any closing remarks.
Thank you again for taking the time to participate in the call. And more generally, thank you for your support of VBI. I would like to finish by saying that, based upon the results shared here with you all today, we truly believe that VBI has the potential to be part of the public health solution by offering a rapid, effective, safe, and differentiated approach. We look forward to the start of the clinical studies around the end of this year and to sharing future data. We are, of course, available to be contacted individually, and we'll be happy to provide any information we are able to discuss. So with that, again, thank you for your support of VBI and our efforts to tackle this huge public health challenge. We'll sign off here. Thank you once again.